Hello, I'm Clare Boyes, and I'm going to talk to you today about my dissertation project, A Bee's Eye View of Landscape Change. And this project arose following a discussion with Stuart Roberts at the last in-person BWARS AGM back in 2019. And Stuart had worked with Tom Wood, who many of you will know, to compare historic and contemporary diet breadth in polylactic Andrena bees. And the historic data was taken from the work of Vic Chambers, and I'll tell you a bit more about him in a minute. And Stuart and Tom found that over time, the diet breadth had actually remained the same, but the proportions of the different pollen families in the diets of the bees had changed. So rosacea pollens, such as hawthorn and blackthorn, had decreased, and brassicaceae had increased. And they attributed those changes to increasing agricultural intensification following the Second World War. So here's Vic Chambers, and he lived and worked in Bedfordshire. And he was an amateur entomologist with a particular interest in Hymenoptera. And these are some of the pages from one of his notebooks. And because his other big interest was pollen analysis, and he detailed those in these little notebooks, which Stuart very kindly lent to me. And between 1933 and 1966, he collected almost 1,300 pollen samples from a number of different species of mining bee, and wrote down his analyses in all these books. Now, the most numerous of the species that he collected the pollen samples from was Andrena flavipes, with nearly 500 samples. And you'll notice here is written at the top, Andrena flavipes, Long Lane Tingrith, 22nd of April 1945, and then sometimes 2:45 to 4 p.m. And that was the time he spent that day collecting a number of samples. The next most common species he collected samples from was Andrena barbilabris. Now, both those species are still fairly common today, and more importantly, they're still present at some of the same sites that at which Vic Chambers found them. So my research objectives were to go along and collect more pollen from these species and analyze them and compare that with the historic data from Chambers, and then to identify any potential links between land use change and the diets of the bee. So the field sites, as I say, he lived in Bedfordshire, so they were quite near Milton Keynes and Luton. You'll see the dots on the map there. Thankfully, they were quite close together, but they were quite a long way from where I live in mid Wales. You'll see the little dot over at Welshpool. So I had quite a long drive, and it was quite tricky second guessing what the weather would be like. The other thing that, of course, was that the pandemic threw a spanner in the works. So I was due to start the field work in the spring of 2020, but in the event, I wasn't allowed to leave Wales until mid-April 2021. But I used that intervening time to build up a pollen reference collection, which was very useful later. So my first field site was Long Lane Tingrith, and the field site's in the centre of that image. And as you can see, it's quite intensive arable farmland. And the bee I was looking for there is Andrena flavipes. Probably many of you are familiar with that bee. And I visited the site 8 times between the middle of April and the end of May. I only found females with pollen on the middle 6 visits. And I was quite surprised because I thought that the season would have been well underway by the time I was able to get there. But on the first visit, though, although there were a lot of males swarming around, didn't find any females with pollen. And over the course of those 6 visits, I managed to collect 30 samples, but it was very slow, and I was averaging just 2 samples per hour. And the bees were nesting in this embankment, and Chambers describes how this was a very short sward in his day, and because it was grazed by rabbits. But that's no longer the case. You can see it's quite coarsely vegetated, and there are a number of garden plants in there, such as daffodils, Spanish bluebells, and red hot pocus, and a Leylandii hedge at the top. I'm sure that wasn't there in his day. And the bees seem to be restricted to nesting in the bottom about 18 inches of the bank, where I think the car tires have kept it a bit clearer of vegetation. And just either side of that aggregation were these hedges. So as you can see, they're not in great condition. And if we look at the land use change, historically, the land around that site was mainly grassland, and that would have been species-rich grassland. And nowadays, most of the land around that site is arable land, so no great surprise. My other site, Aspley Heath, is no longer a heath. It was forested in the late 1700s for timber with mainly Scots pine, but some deciduous trees as well. The bee that lives there that I was looking for is Andrena barbilabris. And if you're not familiar with this bee, when she lands on the sand, she will then swim down through the sand to her nest burrows. And you can't see where the burrows are. So it's quite incredible to watch. I visited that site 12 times between mid-April and the end of June, and I only found females with pollen during June, and that surprised me again because I thought I was a bit late. I might have missed things, but I was looking in the right place. I just didn't find any bees until that point. And the bees were nesting in this well-worn track that runs through the middle of the site, and it was very busy because people were still on furlough, so there were a lot of runners and walkers and dog walkers and cyclists and horse riders all going up and down, and also a lot of heavy machinery, because just beyond the aggregation, they were clear felling an area. So the area on the right was being clear felled last spring when I was doing my field work. The area on the left was the other side of the aggregation, and that had been felled maybe a couple of years ago. But you'll see from those images, there's very little ground flora evident. There was very little flowering at that site. And when you look at the land use change, it's a lot less stark. So it's still mainly woodland. Maybe there's been a small decrease in woodland and a small increase in grassland, although it might be an artifact of the fact that they use satellite imagery. I don't know. So how did I collect the pollen? Well, I waited till the bees were returning to their nest laden with pollen, caught them in a net, popped them in a glass tube with some crushed laurel leaves. And then as soon as they began to lose consciousness, I could tip them into a clear vial. Then pop that in my dark bag and leave them for a few minutes to recover. And then as they recovered, they would clean the pollen from their body and I could release the bee. Now, interestingly, Andrena flavipes took a lot longer than Andrena barbilabris. Barbilabris females would be ready to be released sometimes within 20 minutes of me catching them, whereas the Andrena flavipes took sometimes an hour and a half. So they were exactly the same methods that Chambers used. And I used the same methods for pollen analysis as well. I counted 1,000 grains of pollen and identified them. And I had my reference collection to help me. And on each visit to the sites, I'd made a list of the plants that I could see and that were flowering. Rex Sawyer's key was very useful, and I digitized that using some software from the Field Studies Council. So I'd got a little computer program that was helpful, especially when I first started doing the pollen analysis. And then for the more difficult pollens, the online resources were particularly helpful, especially this one called Poldat, which has thousands and thousands of images, and it's searchable on the different features. Very, very useful. So Andrena flavipes, and what did I find? Well, obviously I had a lot fewer samples than Chambers, and contrary to wooden Roberts, I didn't find a decrease in rosacea and an increase in brassicacia. It was the opposite, but that wasn't significant. And really, the significant changes were in Asteraceae pollen, and that was sycamore in the current samples, and there was a sycamore tree just opposite the nest site. Chambers had found that the bees actually used field maple, Acer campestre, but you will have seen from the slides of the hedges, the field maple wasn't in any fit state to flower. And Asteraceae pollen had increased, and that was mainly dandelions, and there were a lot of dandelions growing in that bank. But the other thing about that the samples is that the numbers of different pollens had decreased. So in Chambers samples, there were at least 25 different types of pollen found. I found 17. And there were 14 pollens that only Chambers found, 11 that we both found, and then 6 pollens that only I found. So a bit of turnover in the types of pollen. And the other thing that was different was that the average number of species per pollen load increased. So Chambers had found 2.4 species in a pollen load, and I found 3.3. So the bees were collecting more types of pollen on one foraging journey. And of course, the colony size had changed. As I pointed out, he'd written the times that he was collecting at the site, and actually he only recorded that for Andrena flavipes, and he was averaging at Long Lane in the spring generation 36 bees an hour, and that was compared to my 2, so quite a difference. And as I said, the bees were nesting at the bottom of the bank, going into these cracks and crevices really, and Chambers had managed to put glass tubes over the entrance to many of the burrows, so that had made it easy for him to catch them. And the other thing that's interesting about Tingrith is that there was always a higher proportion of rosacea pollen in those samples compared to the other samples in Chambers' data sets. So the other samples in his data sets averaged about 20% rosacea pollen, whereas Tingrith, it was about 40%, and it increased slightly in my samples. And the area I've circled is an old orchard. So it had flowering plums and apples when I was there. And I suspect that orchard has been there for 80 years at least. And I think it's probably always been quite important for those bees. Now, Andrena barbilabris, because there was so little ground cover, I thought that the pollen analysis would be a lot easier and more straightforward. And the first sample I looked at had me stumped for a while, and it turned out to be a Fagaceae pollen sweet chestnut. And I had seen that flowering on the heath, but it was quite some way from the nesting aggregation, so I'd kind of discounted it. But the pollens collected by Andrena barbilabris were really quite different from in Chambers' day. So rosacea pollen that had decreased significantly, as had Asteraceae pollen. Cornacea had increased significantly, and that's dogwood, and that formed the largest proportion of the pollen grains. And dogwood was not growing on the heath, and I confirmed that with the rangers, but I think it was growing in the gardens nearby. Brassicacia pollen, that stayed about the same. I think that was growing in a vegetable garden nearby. The Apiaceae had completely vanished. There were no species from that group found. Chambers had found 4 or 5 different species, but there was nothing like cow parsley or hogweed apparent anywhere. And Fabiaceae is broom pollen, and that was new pollen that had not been found by Chambers, but nearly a third of the pollens were from that plant. So a big change, and the diet breadth had decreased quite a lot. So Chambers had found 29 different types of pollen in his samples, and I found only 13. So there were 21 pollens that only Chambers found, 8 that were common to both of us, and then 5 novel pollens. So quite a big difference. And if we look again at the satellite imagery of the heath, I've put some buffers on there. And that inner circle at with the 500-meter buffer, you can see the areas of clear fell that I've shaded in orange, and that's about 10% of that middle portion, and the yellow dot being where the nesting aggregation are. And then I've mapped out the sweet chestnut trees with the asterisks or the stars with information from the NBN gateway. So it might not be exactly accurate, but it gives an indication. And I certainly didn't see any close by. So I think the nearest sweet chestnut trees were about 700 meters away. And I think the gardens at the top left of the site were particularly important. So I think quite a lot of the pollens came from there. And there was some exotic pollens mixed in small numbers, but things like lilies and Nortzia, which lends weight to the idea that they were foraging in those gardens. And that was interesting because there's one paper that said that foraging distance is governed by the size of the bee. And so they'd estimated that if you translocated bees and if you translocated Andrena barbilabris from their nest, then only 50% of the females would return from a distance of 120 meters. And if you moved them to 530 meters, only 10% of the females would return. And that's really at odds with what I found here. And there was another paper by a lady called Beale who found that she did something similar. So she mapped the pollens found on the bees with the pollens in the landscape and estimated much longer foraging distances. And certainly 27 of the 30 samples I had contained at least some pollen, a proportion of pollen from at least 600 meters away. So they were flying a long way. And the other thing that changed for both species was an increase in the proportion of pollens that came from woody plants. And Andrena barbilabris, it was already a high proportion, so it was already 3/4, and it increased to almost 90%. And the only pollens that weren't from woody plants were actually the brassicaceae pollens. And Andrena flavipes had a small increase, but still significant, to 65%. So looking at the diet breadth, I used a program called INEX, which calculates this thing called child species richness. And that takes account of the fact that I had different sample sizes. And what it's doing is saying that if you kept on sampling it, you plug in your results, kept on sampling until you find no new pollens, then in the 1940s, there'd have been 43 species that Andrena flavipes was using. And in 2021, there'd have been 30. And for Andrena barbilabris, those numbers were 42 for the 1940s, so very similar, and 30 for last year. So both species showing a decrease in diet breadth. So yeah, that was my conclusion. The diet breadth of both bees decreased, and there was an increase in pollens from woody plants. And Andrena flavipes was collecting more types of pollen in a single load. That wasn't the case with Andrena barbilabris. It was still it collected an average of 2 both periods, but it was flying long distances. Whether that was longer than it used to be, we can't know. It's probably linked to the land use changes. So I think for Tingrith and the Andrena flavipes, that's the intensive agriculture. But it was more surprising, the changes at Aspley Heath, which I think maybe reflects disturbance on the site, maybe the pandemic pressures. I saw something similar the other week on Hampstead Heath in London, where a lot of the ground flora has disappeared for the moment anyway, because it got such a hammering during the pandemic with people using the area for leisure. Perhaps the new agri-environment schemes may give us an opportunity to plant some trees that would benefit pollinators. There are a number of schemes in there that focus on tree planting and tree cover. And it's interesting that the bee's eye view highlights the changes which are not immediately apparent at the landscape scale. And it just remains for me to thank Stuart for suggesting the project and supplying access to Chambers notebooks and digitized data, and my supervisors, Jennifer and Emma, and particularly Liam Crowley for encouraging me to believe that I could actually do pollen analysis. So thank you. And Andrew Green and the Green Sand Trust for permission to carry out the sampling on Aspley Heath. So thank you very much for listening. And if you've got any questions, I'd be happy to take them. Thank you.